Good morning, everybody. Today is Wednesday, June 29th, 2016, and this is Before the First Cup with Jules here on UCY.TV. Happy Wednesday, everyone. It is an overcast 66 degrees, a little bit damp and chilly here in upstate New York. We had some rain last night, which was pretty nice, and hopefully the plants all get perky. So where did we leave off yesterday? We titled the show... Oh, let's not have one of these shows now. Hold on. Everything is moving like syrup. I'm there. What do we title it? I don't know. I never remember what we titled the show. We titled the show New Gene Editing Cures Equals Psychopath-Directed Biological Evolution, and that's exactly what it is. You listened to that interview 2 days ago by Harvard geneticist George Church talking about the augmented human being and all the wondrous things that we could do with CRISPR technology, being able to splice and dice humans, knock out genetic diseases, add all kinds of wonderful new traits potentially in the future. He wrote a book that's out on Amazon titled How Synthetic Biology Will Reinvent Nature and Ourselves. It talks about that very thing. We discovered also that he is involved with the woolly mammoth cloning project. He is also involved with the brain project. He is involved with CRISPRing the first humans, and he is involved at Harvard University with them deciding to create the first synthetic human genome. Good old George Church is quite the connected individual. He helped co-discover the Weiss Institute for Biological Innovation, whatever they call that, biologically inspired engineering, stealing biology secrets to turn them into technologies that humans can exploit. We're going to continue a little bit more looking at CRISPR technology. I had a story where we ended yesterday and we didn't quite get there, but we've been given now the green light to start CRISPRing humans. There's just a few little ancillary approvals that they need to get. One, of course, is from the FDA, but I don't think that's going to be very difficult. The FDA seems to be pretty slippery, and I actually found a video about the revolving door between Big Pharma and the FDA that if we can get it to work later, maybe we can listen to it. It's Ben Swan, but I remember when it first came out, it's actually very good. But yeah, so they've given the green light just about to CRISPR people. Going to be looking at doing that at the University of Pennsylvania, of course, in the name of curing cancer. There's no guarantees, of course, but don't worry about it. Step right up. 21st century medicine. And we are going to give you the benefit of potentially being the very first human who could be gene edited and made better. Right? That's the way it's going to be. They'll be able to fix every error that we have in our genes in the future because we are defective humans. And of course, you know, that isn't for you or me. Like I always say, it's because we are the free R&D that then gets version 1.0 into a version 4.5.0, so it's perfected to then be used on those who have all kinds of cash invested in this to allow them to be perfect, superhuman individuals that never die. And, you know, beyond that, once they've been able, and we discovered listening to George Church, he said, you know, not only can we CRISPR humans, but we have the ability now to CRISPR everything on the planet. And remember, CRISPR, when it was first being created, was just a killing machine in the beginning until they discovered how to zip-zap genes, unzip, insert, rezip. One thing I wanted to see here, and I do, here, everything we need to know about CRISPR, but we're afraid to ask. I don't know. This computer is like doing some really weird shit this morning. 5 minutes ago it wasn't. Everything was great. I had the whole show open and ready to rock and roll. While this is opening, I saw an interview yesterday with Stephen Hawking. We've looked at him quite a few times. and talking about artificial intelligence and like how damn crazy we are for wanting to develop an intelligence that is not only smarter than us, but who can evolve because we're going to be in big time trouble. Well, Stephen Hawking comes out in this interview with Larry King the other day. People ever more greedy and stupid will destroy the world. And I think if there ever had to be a title, and I say this pretty often, but if there ever had to be a title of the gist of what this show is about, I would have to say that's about it. It's greed and stupidity of the human race, bringing us to our knees and destroying it all. I mean, who knows how many times this has happened? I'm almost going to guess that as scientists tell us that we are at the brink of the sixth great extinction, pretty much anything goes now, because what does it matter? You know, they're thinking. And one of their big things is they want to get off this rock. I've seen a lot of news about Mars, growing plants on Mars soil. I just saw some scientists are messing with now. They discovered that Mars had tons and tons more water than it does currently, just like in Earth. They're working on the first government for Mars, and they're still talking about that moon base. So put on a seatbelt because it is coming. All right, anyway, let's start here. For people who don't necessarily listen every day, I hate to like jump right off into the deep end, because if you've never heard about this technology before, you might be scratching your head going, Jewel, I just don't get it. So we're going to start with a Gizmodo story. This one's from May 2015, actually. So a little bit before, I'd say just over a year ago, where we were still learning what the acronym CRISPR was all about. Everything you need to know about CRISPR and the new tool that edits DNA. So this is when editing humans was just a glimmer in some crazy scientist's mind in a dark back room, rubbing their hands together. CRISPR, a new gene editing tool, could transform the field of biology, and we've gotten to that point just one mere year later. In a recent study on genetically engineered human embryos, which they did have permission back a year ago to tinker with in the UK and in China. And just this spring, they approved being able to tinker with human embryos in the United States. Has converted this promise into media hype, but scientists have been tinkering with genomes for decades. So why is CRISPR suddenly such a big deal now? George Church said CRISPR just came about in 2012. So that's a very good question. It's pretty insane to me that in just a mere 4 years, we have gone from a petri dish lab experimentation to literally looking at CRISPRing human beings who are alive and functioning to see what's going to happen. The short answer as to why CRISPR is such a big deal is that CRISPR allows scientists to edit genomes with unprecedented precision, efficiency, and flexibility. And the past few years have seen a flurry of firsts, never before seen. Remember, too, we found out back, 6 months ago or so, there was a not-for-profit company called Adogene who sent 60,000 CRISPR editing kits to whoever wanted them all over the globe. So we could have little Johnny DIYing gene editing in mommy's basement in Pakistan. I don't know if they have basements in Pakistan, but you know what I mean. And doing whatever they want with this stuff. If this is so precise and just so earth-shattering and groundbreaking, why are we democratizing the editing and creation of life? Like, whose bright idea was that? It almost seems to me like it was a plan to create mass confusion. So you know what? Problem. Ding. Problem. Now we have genetically modified organisms we've never seen before. Pandemics running rampant. We're going to look. I was reading a story today about some clinical trial hoopla that happened at NYU. I guess there were 8 studies that got shut down because it was considered, I don't think it was unethical, but they definitely weren't following the rules, and some some administrator who stepped to the plate for 5 minutes with a backbone of ethics shut the experiments down. And they discovered that no one was doing follow-up on any of these patients, that they were giving this quote-unquote marijuana-like substance to treat PTSD. Sounds a lot like some of the original experiments that were happening actually with cannabinoids that Dinah used to talk about, except now they were using a marijuana-like substance, it said. So some sort of synthetic pot that they were going to see if they could treat PTSD, and they were feeding it to these people, and no one was checking up on them. Now, that shows how much. Step right up for your, I don't even know if they paid them. Found out that GMO banana, Bill and Melinda Gates, GMO banana with the added vitamin A at Iowa State University, they were going to pay the college kids $900 a piece to eat one banana. First ever time tried in humans. Makes you wonder why they would pay for that. It makes you wonder why they need human guinea pigs. Not really. Anyway, so why is CRISPR such a big deal? Well, we're going to tell you. The short answer is that CRISPR allows scientists to edit genomes with unprecedented precision, efficiency, and flexibility. And just these past few years have seen a flurry of firsts with CRISPR, from creating monkeys with targeted mutations and to preventing HIV infection in human cells. Now, it's interesting they mention that because the Chinese were messing with that over the last year, and they said that it didn't work. So this article is from May of 2015, so that might have been before the Chinese came out and admitted their failure. It says earlier this month, Chinese scientists have announced that they applied the technique to a non-viable human embryo, hinting at CRISPR's potential to cure any genetic disease. And yes, this even might lead to designer babies. And we talked about that for Price Man, Johnny Knibb. Brown hair, red hair, blonde hair, big nose, little nose, blue eyes, green eyes. Pick them. Spin the wheel. Let's see what happens. In short, CRISPR is far better than older techniques for gene splicing and editing. And you know what? Scientists didn't invent it. No. CRISPR is actually a naturally occurring ancient defense mechanism found in a wide range of bacteria, and as far back as the 1980s. Again, the 1980s, guys. Though I still love the 80s because that was my coming of age. That was in my... Yeah, actually, it was my coming of age. I hit 14 in 1982, and I hit 18 in 1986. And the 80s were good for me. I enjoyed the 80s. Lots of crazy teenage years. But you know what we didn't realize? Even look at back in the music. You know, I was into all that new wave weirdo shit music. And it was all about, you know, the first synthesizer, the first recording VCR. We first got cable. It was, we first got our first video games. I got my first Commodore 64. The 80s were all about the technological revolution. The 80s were about moving into the future and the promise of what it could mean. The 80s brainwashed people of my generation to believe that where we've ended up today was going to be a utopia. There would be no more war, famine, and the world would be a happy place. And we will be able to push a button and get our lawn mowed. And I mean, we're just about there, people. But what we didn't see back in the 80s was the dark, nefarious side to all of this, the manipulation and control of the globe that now is going to move to a cellular and DNA level to be able to manipulate and control every living organism on this planet. And that's what CRISPR's all about. All right, let's get back to this. So as far back as the 1980s, also when they first started messing with genetically modified foods and vaccines in foods as well, scientists observed a strange pattern in some bacterial genomes, and one DNA sequence would be repeated over and over again with unique sequences in between the repeats, and they called this odd configuration clustered, regularly interspaced short palindromic repeats, or more affectionately known as CRISPR. CRISPR. This was all puzzling until scientists realized the unique sequences in between the repeats matched the DNA of the viruses that they came from, and specifically viruses that prey on bacteria. Turns out CRISPR is one part of the bacteria's immune system, which keeps dangerous bits of virus so it can recognize and defend against those viruses next time they attack. The second part of the defense mechanism is a set of enzymes called Cas, short for CRISPR associated proteins, which can precisely snip DNA and slice the hell out of invading viruses. Conveniently, these genes that encode for Cas are always sitting somewhere near the CRISPR sequences. There are a number of Cas enzymes, and the best one known is Cas9. This comes from a streptococcus, better known as the bacteria that causes strep throat. And together, they form the CRISPR-Cas9 system, although it is also often just shortened to CRISPR. So anyway, at this point, you can start connecting the dots, something that we've done on our own. Cas9 is an enzyme that snips DNA, and CRISPR is a collection of DNA sequences that tells Cas9 exactly where to snip, and all biologists have to do is feed Cas9 the right sequence, called a guide RNA, and boom, you can cut and paste bits of DNA sequence into the genome wherever you want. DNA is a long string of 4 different base pairs, A, T, C, and G. We've talked about that a lot, and other enzymes used in molecular biology might make a cut every time they see, say, a TCGA sequence, going wild and dicing up the entire genome. CRISPR-Cas9 system does not do that. It only recognizes a sequence about 20 long, so it can be better tailored to a specific gene. All right, so I don't know if we want to really get into this. Is a really long story, but I think that gives us a good understanding of where the CRISPR comes from, where the Cas comes from, where the 9 comes from. They're using streptococcal bacteria. That's pretty interesting. I did not realize that. So now they're doing this in humans. They're going to be sucking out T cells out of the immune system at the University of Pennsylvania, editing these T cells to not turn off, I believe, is what we first were reading. And then they're going to inject those genetically modified T cells back into the cancer patients, hoping that the T cells, now that they are in an always-on mode, will be scanning and finding all the foreign invaders that it missed, like the cancer, to be able to overwhelm that cancer and kill it. And remember, we found out there's all kinds of dangers that are associated with this. We've talked about autoimmune disease, and one of the things that happens in autoimmune disease is that your body incorrectly identifies parts of itself as foreign invaders. And I absolutely could see this method that they are talking about using with T cells to turn the off switch off and just let them go awry in the body as being a potential to some disaster. In this magazine called Stat, this is Stat in the Lab, there was a story that came out the other day called "They Are Going to CRISPR People and What Could Possibly Go Wrong." It's from June 23rd, 2016, so 6 days ago. I'd like to share this one with you. Now that we know all about CRISPR and all about what they're proposing to do, well, we've postulated all the crazy things that could happen, like arms growing out of the back of your head. Well, let's find out what they say here. And we also have, don't forget, the Bill and Melinda Gates Foundation, Google Ventures, and Facebook billionaire Sean, and Napster, I keep forgetting that one, Sean Parker's money involved in this research and this R&D. All right, to this story. They're going to CRISPR people. What could possibly go wrong? Well, now that Federal Biosafety and Bioethics Committee has approved what would be the first use of the trailblazing genome editing technology, CRISPR-Cas9 in people, the obvious question arises, could anything go wrong? As Homer Simpson sits there and scratches his head. Well, the purpose of such a phase 1 clinical trial, yeah, for all the dummies that get involved, is to assess safety. So problems would not come as a total shock, right? Because phase 1 means we're going to try and work all the kinks out. And the fact that the trial in cancer patients, which of course still needs the okay from the FDA among others, would be funded by the new Cancer Institute founded this year by Sean Parker, Facebook mogul, also adds a wild card to this picture. And they don't even mention that the project itself had also received Bill and Melinda Gates funding as well as Google Ventures. There are 4 potential snafus. 1, CRISPR edits DNA it isn't supposed to. Whoops. Soon after scientists reported in 2012 that CRISPR can edit DNA, experts raised concern about quote-unquote off-target effects, meaning genes that scientists didn't intend to change inadvertently, got deleted or altered. And that can happen because one molecule in the CRISPR system acts as a molecular bloodhound, sniffing around the genome until it finds a match to its own sequence of A's, T's, C's, or G's. Unfortunately, in the 6 billion such letters of the human genome, there can be more than one match. I didn't know that. And the proposed CRISPR experiment, which would be led by scientists at the University of Pennsylvania, would use 3 of these molecular bloodhounds, tripling the risk of off-target effects. The experiment wouldn't alter the immune system's T cells. The experiment would alter the immune system's T cells only after they are removed from the patient. And yes, we mentioned that. So that gives scientists a chance to screen the CRISPR cells to make sure that only the 3 intended genes, all involved in making T cells, find and destroy tumor cells, are altered. But after those T cells are infused back into a patient to fight melanoma, sarcoma, or myeloma, the CRISPR system can keep editing DNA, and tracking these edits become like following a polar bear in a snowstorm. Now, I didn't know that. So CRISPR, once they set it loose, even if they suck those T cells out of your body, edit them and put them back, CRISPR keeps editing. Like I said, George Church in that interview, the augmented human from 2 days ago, had said the original CRISPR was a killing machine. I don't think I'd be putting this into my body, even if I were desperate. All right, so this continues. Dr. Michael Adkins asks, how will you identify off-target editing of the Georgetown Lombardi Comprehensive Cancer Center, a member of the Federal Recombatant DNA Advisory Committee that approved this experiment? Yes, they are part of that group where the FDA kind of bowed out. They don't have to approve now every single case of human gene therapy, but they're going to allow this Recombatant DNA Advisory Committee that's existed. I'm trying to remember, I think it was the 80s again, and we discovered, maybe it was even the 50s. It was back around Plum Island time that Recombatant DNA Advisory Council Committee had been formed. But they now are picking and choosing which cases to review. So not even every bit of gene therapy, quote unquote, zipping and unzipping genes in humans is going to be reviewed by them. But this particular case, the very first one in CRISPRing humans, was. Anyway, Penn University's data on cells growing in lab dishes show off-target effects of 148 genes that they thought might be inadvertently hit by CRISPR, and only ANK1, a gene that is active in the brain and the blood cells, was hit. But we can't be sure that this will be maintained over time, says Atkins, meaning whether CRISPR will edit the wrong DNA once the T cells are back into the patient. Hitting the wrong DNA targets in gene therapy experiment has also caused tragedy in the past. In 2002, a little boy developed leukemia after a gene he received landed on a spot in his DNA that activated cancer-causing DNA. Whoops. Scientists have made tremendous progress in minimizing CRISPR's off-target effects, says Dr. J. Keith Jong of Massachusetts General Hospital. which is involved in that whole Broad project, the Broad Institute, I believe they are called, another organization that ties Harvard and MIT, but this also ties a lot of the universities and the hospitals together and involves who? Oh, good old George Church again. But some of the progress that has come from benching CRISPR's standard DNA cutting enzyme and substituting one called Cas9, as Young's team has reported, and the Penn team is not using this HF1. Penn's Dr. Carl June, not Carl Jung, Carl June, who will lead the study, told Stat, this publication, that CRISPR science is rapidly evolving. So they will be using the state-of-the-art technology at the time the study opens. Phew, thank God. New and improved. And at this point, we have compelling preclinical data with standard Cas9. All right, so that's number one. So we can hit off target genes, but don't worry about it. We think we're good. Even though it keeps CRISPR editing once it's inside the body and it's like tracking a polar bear in a snowstorm, don't worry about it. You'll probably have to have some sort of little wireless monitoring nanobots or something inside your blood to keep track of those. Who knows? It's insanity. All right, number two. Second, what possibly could go wrong CRISPRing humans. Number two, CRISPR hits the target, but then all genetic hell breaks loose. Uh-oh, that would be a problem. When CRISPR's DNA cutting enzyme snips the genome, the severed DNA strands don't just smoothly reconnect like an electronic document that closes up the space between just and reconnect if smoothly is deleted from the sentence. No. Random DNA floating around rushes into this gap. Well, if the DNA equivalent of, say, politically, the word, or misanthropically, the word, scoots in to the genetic quote-unquote sentence, the results could be as nonsensical as putting those adverbs into that sentence above. The problem is serious enough that scientists are racing to solve it, and no one knows the biological consequences. Oh my God. No one knows the biological consequences if Penn's system doesn't. So only a safety study like the one Penn is proposing can determine, and that is, safety study means phase 1 clinical human trials. So these poor people, I hope they realize this. Only a safety study like the one Penn is proposing, quote, can determine if there is acceptable feasibility and safety of this approach, unquote. And I mean, to me, everything that I've read, Penn is ready to chomp down on this and get running. This is not like a maybe. They already got the green stamp approval from that recombatant DNA committee, and now they're just waiting for the FDA and some hospital to step right up and say that they will be involved in the testing. I bet you that Massachusetts hospital will. They're part of that Broad Institute. They're part of this same group that's all going to profit from splicing and dicing you and me and Nana and Johnny. It's just, oh my God, this bothers me so much. Anyway, but since all the study volunteers, remember, step right up and give it to the machine to be able to help all of humanity, because that is your duty as a human. George Church had his Genomic Institute that was saying that we should be doing that as well to help them find cures for cancer. The National Institute of Health is doing it through 23andMe to help us do a 1 million person study to become a precision medicine cohort to help cure diseases worldwide just by giving it up. But since all the study volunteers will have incurable cancer, these risks, quote, would be acceptable, unquote. What did I tell you? Be a total guinea pig. You're going to die anyway, Johnny, Mary, Susie. So step up. Let's do some freaky mad science. Gone mad on you. All right, number 3. What could possibly go wrong CRISPRing people? And there's only 4 of them, I believe. I don't know. That's what I saw yesterday. Yeah, there's only 4 of them. Number 3, the Energizer bunny problem. Remember how annoying that Energizer bunny was? The components of CRISPR usually don't just slip into T cells on their own. That requires a virus, since viruses are adept at infiltrating cells. We've talked about that. The reason why they've been using viruses, not just in CRISPR, but back in genetic modification of plants and animals. That's how I discovered watching a documentary on GMO and how they made a freeze-resistant tomato. They used a bacteria to CRISPR in. I don't know if they actually CRISPRed it. It might have been like the old school GMO. Was it Talon, they said? To be able to splice and dice, let's put it that way, because it's all the same insanity. They used a virus where they sucked out the guts of all the viral material and they inserted into the guts some sort of benign instruction set along with the DNA from an Arctic flounder, I believe it was. The exact genes that prevent that Arctic flounder from freezing in the cold Alaskan waters. And they took that, they put it into the virus. They put the virus into the tomato. And I'm assuming it was an embryonic tomato. I don't know. They didn't say if they were doing it on a live grown tomato, if they were starting from scratch. But regardless, the tomato genes, they put that into the tomato with the virus. And what the virus did was scan the cells and they look for the chinks in the armor, right? We've talked about that before. That's how genetic modification works. It is a Trojan horse that invades and circumvents the quote-unquote immune system of the organism. I mean, anything that does that cannot be good. It circumvents millions of years of evolution, protecting our genetic material that has allowed us to become what we are today. And it's going to circumvent that, bastardize it, and turn it into whatever the mad scientist is trying to unnaturally force into that organism. That's wrong. That's just so wrong on so many levels. All of it. It puts them in the position of gods. They're creating life in their own image, in their own crack-potted mind as to what they want to see. And don't think that this is even the end of it. Remember, I keep saying this is only the beginning. Think of the horror if you just let your mind just wander with the possibilities once they're able to like completely knock all the hiccups out of this technology. Think of all the horrible things that they could do. Think about like the flying creatures with x-ray vision and fangs and big pointy teeth. It's just beyond comprehension. We're worried about robots right now. I think I'd be more worried about clone genetically modified Isle of Dr. Moreau creatures. Anyway, number 3, the Energizer Bunny problem with what could possibly go wrong CRISPRing humans. The components of CRISPR usually don't slip into T cells on their own. That requires a virus, since viruses are so adept at infiltrating cells. And a spokesman for Penn University said that the scientists were not available to answer questions about their proposed procedure. But if they did use viruses, they run the risk that virus-infected cells will keep cranking out DNA snippets of Cas9, and by one estimate, for another 10 to 20 years after the experiment. What? That leaves a lot of time for unintended genome editing to occur, since it just continues on inside the body for 10 to 20 years if they use the technologies that they've been using up to this point. And number 4, of course, what could possibly go wrong with they're going to CRISPR people? Dollars triumph over data. Ding, 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 ding. We got rich tech billionaires. And everyone is clamoring to get their name in the history books as the one who invented this or that, because that makes them filthy rich and elevates them, at least temporarily, to the god science caste that has evolved in this technocracy that we call, I don't even know what we call it. It's more than the United States. This is the New World Order. Science is going to be able to manipulate and control everything. And those in the know, those who are investing in the technology, rich tech billionaires. DARPA, military industrial complex, is also investing and helping them to further their research, giving them soldiers to be able to experiment on. We have our federal government stamping out initiatives left and right. Got precision medicine initiative. Now we got the microbiome project. We got the brain initiative. Woo! It's going to make a great 21st century economy as they sold us to big pharma to be the guinea pigs. But let's get rid of that pesky FDA. And again, that FDA is standing in the way of these 18 people who are about ready to take part in this human CRISPRing trial. It is standing in the way of them potentially finding a cure. I mean, like I said, that hospital is going to go for it. I bet you it'll be that Massachusetts one that's part of the Broad Institute. But that FDA has to give approval. So will someone on the FDA board get a backbone, or are they just going to kiss ass with Big Pharma and Big Agra and the feds and the mad scientists and the military industrial complex and lots of rich tech billionaires wanting to cure death as a disease? Are they going to stand up and not rubber stamp this? I'm going to have to guess probably no. I think they're all a bunch of jellyfish. So dollars triumphing over data. Number 4, what could possibly go wrong with CRISPR in humans? Well, if Penn's experiment goes well, of course, larger clinical trials would follow. But, quote unquote, well is subjective. Measures such as whether a patient is alive or dead are, of course, tough to fudge. How bad side effects are less so tough to fudge, and judgment calls can even enter into assessments of how significantly and for how long tumors have shrunk. Study after study has shown that when clinical trials involve entities with a financial interest in the outcome, ding, such as the Parker Institute for Cancer Immunotherapy and Penn University have in this one, and they do. There's big money riding on all this. The reported outcomes are more likely to be favorable than when the trial is sponsored by, say, the National Institute of Health, which I would even question that one. I would say if it was some small university lab who finds out that GMO foods cause grapefruit-like sized tumors in rats, those would be way easier to debunk. Point 2 is you got billions of dollars behind the technology going, that's a bad study. But when you got billions of dollars behind this and all these investors, and we've spun Adidas Medicine, licensed CRISPR technology to them, and then spun Adidas Medicine out with, again, funding by Google Ventures and Bill and Melinda Gates to go on an IPO in January 2016. There's a lot of famous and rich people who bet the farm on this. And you better believe that FDA is just going to play along, slip enough cash in there and pat enough butts. And I don't think you're going to have to worry about anything. We read some stories, too, that there are prominent scientists around the world standing up, talking about the crisis we have in published research right now. I believe that they'd said more than 50% of the research out there currently is potentially fraudulent. And it was all fueled by cash. You can't trust anything that you read right now, especially if there were big bucks and big organizations behind it. So this is beyond frightening. This is the ultimate in hubris. These people are going to make it happen whether you want it to happen or not. And there are no safeguards. There are no breaks. If they want to make it happen and the regulatory agencies get in the way, well, then we just do it in the closed back room and we don't tell anyone until we have a breakthrough. And then it'll be this miraculous discovery that came out of nowhere, just like CRISPR in 2012. Anyway, in studies where the sponsor has a profit motive, Scientists are also less likely to adhere to best practices. Wink, research has shown. So, if you really believe in a biotechnology and it is not completely clear whether or not a side effect is the fault of the disease or the technology, your bias could influence how you interpret that data. In 1999, members of the Recombinant DNA Advisory Committee pointed out a young man died in a now famous gene therapy trial where, at Penn University, in which the lead scientist had a multi-million-dollar financial stake in the technology, and that conflict of interest, scholars have argued, may have led him to make dangerous decisions. And although the Parker Institute will handle patents for any discoveries that emerge from the research it funds, I forgot to mention that. That was the one thing I read in another story. So this tech billionaire that came from Facebook as well as funded Napster, this Sean Parker that is funding this CRISPRing humans out of Penn University through his Cancer Institute. What's unusual about this, unlike other cases, is that he is holding the rights for patents for any discoveries that come out of this first ever human clinical trial of CRISPRing. And what they typically do in the past is that it's kind of out there. So when they make a discovery that is like earth-shattering and groundbreaking for humanity, typically they'll allow the discovery to be out there for other labs to try to replicate it, right? It's an open patent system. You might be able to patent, let's say, the technology, but the cures that you come up with tend to be out there for everyone. Well, I guess not in this case. Although the Parker Institute will handle patents for any discoveries that emerge from the research it funds, each site owns its intellectual property, says Chief Legal Counsel Melinda Griffith. If you invent it, you own it. So maybe they clarified that. Or, if everything goes well and CRISPR cures cancer, maybe, just maybe, we'll all get lucky. So I don't know what you want to say about that. It's not like we didn't learn. I mean, I guess we did learn some new stuff. I didn't know that CRISPR could keep editing inside the body for 10 to 20 years after the fact. That is quite insane. What is going to happen when these people turn into a gelatinous mass? I mentioned that yesterday, of just skin and messed up bones on the floor, and they can't fix it. I just envision the worst horror that you could possibly imagine. Do not let this mad science inside of your loved ones. At least not in version 1.0. I just don't see how that is even possible. All right, we're going to attempt, and we're already having difficulty here. I really think that Google must open up a microphone or something when they know I'm talking about something and I'm ready to go somewhere, and they just screw with me. All right, we'll see. Maybe we'll get lucky. Let's see if we can even get Skype to connect. I know. Why do I even try it? This is like Lucy and Charlie Brown. It happens every single time. Hopefully I don't have anything playing on my desktop. I gotta find Jules. There she is. All right, add call. Maybe it'll ring. Hey, we're getting lucky. All right. I gotta unplug my speakers. That stopped the echo, which is good. All right, let's see if we can play this. This is from Truth and Media, which was Ben Swan's publication. I don't know if he's actually doing that anymore, or if he is now. Someone had told me that he's now at a local news station down south somewhere. But we're going to see if my mouse. Hello. Let's be responsive. Truth and Media, revolving door between the FDA and Big Pharma. Let's take a look about how much that's this one protection between you and CRISPRing humans, that is left pretty much. And how much you should be hoping they're going to protect you.
Alone conjures images of collusion, big money, and major influence. In this series, we're going to take a look at Big Pharma and its powerful influence over media, doctors, and even you. And we're going to give you an unprecedented look at the truth behind the tactics, the strategy, and the revolving door between the FDA and these big pharmaceutical companies that claim to be looking out for your health. The first step toward truth is to be informed. And stay tuned after our episode for an important message on health. Well, it is a stunning accusation. The former commissioner of the FDA accused, along with her husband, of making hundreds of millions of dollars while suppressing information about deadly drugs. The RICO lawsuit was filed April 11, 2016 in the U.S. District Court in Washington, D.C. on behalf of 8 plaintiffs who claimed they suffered severe harm by ingesting the drug Levaquin, whose deadly risks were concealed to protect financial interests. The 2 of them are accused of collusion, conspiracy, and racketeering alongside the pharmaceutical giant Johnson & Johnson to conceal the deadly risks of Levaquin. From the lawsuit, while defendant Hamburg was FDA commissioner, her husband, defendant Brown's annual income, not coincidentally, increased from a reported $10 million in 2008 to an estimated $125 million in 2011, and an estimated $90 million in 2012, due in whole or in part to the defendant's racketeering conspiracy to withhold information about the devastating, life-threatening, and deadly effects of Levaquin.
I'm going to stop this for a second because Levaquin, this was from April and I totally missed this, but Levaquin was that antibiotic that almost killed me. I might need to pay attention to this. I hope there's a class action lawsuit. Holy shit, that stuff is like majorly dangerous. When I called my doctor, I was having such horrible side effects. Felt like my head was going to explode. It would make it pound and pound and pound. And she's like, oh, you better stop taking it. It sounds like you're going to have a stroke. The only way I could take it is when I was sleeping. And I think this is also the stuff that screws up all your muscles in your leg. And of course, ever since then, I've had this problem with my Achilles tendon. So let's find out if we can get Ben to play again. Uh-oh.
The lawsuit brings to light just one portion of the problem between troubling relationships in the FDA and Big Pharma. Though clearly what is alleged here, if true, would be one of the most dramatic examples in history. A public citizen petitioned the FDA in 1996 and again in 2006 to issue black box warnings for tendon rupture and tendinitis.
It is.
Warnings been issued. The death toll from Levaquin, reputedly more than 5,000 by the way, and the tens of thousands who were debilitated with life-threatening diseases would likely have been averted. And yet Levaquin was a blockbuster drug, and it was heavily prescribed by doctors. The FDA and many of these organizations that are supposed to oversee these pharmaceutical companies and these drug makers, were they failing in their role to be able to determine whether or not these drugs were dangerous? Doug Bremner is an author, a doctor, and a professor at Atlanta's Emory University. When the FDA is looking at a drug, they're essentially determining whether or not that drug is safe based on the information they're being given. But if the drug itself is dangerous after the fact, they're not following up to determine whether or not that's true?
Congress has limited the budget of the FDA over the years, so they've capped the budget so that it can't increase, but they've also demanded that they increase the speed with which they approve drugs. So the number of people working on new drug approval has been increasing at the expense of those who are supposed to be watching drugs after they come out on the market.
The FDA is approving drugs, but once they're on the market, there's very little oversight, as in the case with Levaquin. But getting drugs past the FDA, that can also be a case of collusion on a number of levels.
The thing about the FDA is not a full-time, a career job. It's a few years on the FDA, and then they're being courted by the pharmaceutical companies.
Lori Powell is a former brand strategist for a medical education agency which serves the pharmaceutical industry. She says there is a difficult balance here because while the FDA officials are supposed to be watchdogs over pharmaceutical companies, they're also constantly courted by pharmaceutical companies.
Right.
We'll have a Merck going in and saying to somebody on the FDA, I'll give you $600,000 if you'll come and work on our regulatory committee to go into bat for us with the FDA. And they'll say, okay, well, I'll think about that. And then there'll be like a Pfizer will come in and say, well, we'll give you $800,000 a year to come in and work on our regulatory committee. And so it becomes like a bidding war like that. So the FDA knows that their next jumping off point to go from a lower paying government job to be a very lucrative pharma job on the inside, the manufacturing side, they know that that's part of their career path. So that, I'm sure, weighs heavily into their decision-making process.
So the question is, should that practice be banned? If you want to work for the FDA, shouldn't that be what you have chosen to do?
Those people that work there should not be allowed to go to work with pharmaceuticals. They should not have any contact with those people. They should realize that their job is serious and it's important. And if they want to work there, that's their career.
And yet fraud between the public and the private sectors happens every day. What you need to know here is that fixing the revolving door between a government agency like the FDA and these massive pharmaceutical companies actually is pretty easy. Shouldn't it simply be illegal to work for an agency like the FDA and then leave that public sector job for a job in the private sector that you once oversaw regulation for? There are some who will say, hey, you can't do that. We're a capitalist economy. But allowing regulators to be in positions of power that can be bought off or influenced by their massive companies that they're supposed to oversee, well, folks, that's not capitalism. It's cronyism. And the only people who likely believe that it's acceptable are those who stand to profit from it.
Dang. All right. And I mean, it's interesting. I did not know about that Levaquin. I need to look into that more because I was a victim of that medication thanks to my doctor. I don't even remember. I think it might have been a sinus infection that she prescribed that for. It was the first time I'd ever taken it. It was a horrible, horrible reaction. So the revolving door between the FDA and Big Pharma, and I mean, Big Pharma is kind of spun out now to the gene editing drug companies like that Adidas Medicine and the new spinoff of 23andMe. Big Pharma is about ready to take a leap into the future, and it's not just going to be pills. It's going to be all kinds of stuff. But if you guys remember, too, there was a woman named Julie Gerbinger, Dr. Julie Gerbinger, that used to head the CDC as well. And we looked at her maybe 6-8 months back. She was the head of the CDC until 2009, after Obama took office. And she was a big part of I'm trying to remember, I always forget what it's called. Oh, it was George Bush with his experimental vaccines in the U.S. stockpile, Project BioShield. She was the head of the CDC during Project BioShield's George Bush days, where they were, post 9/11 and getting all kinds of mad science gone mad, anthrax drugs and universal flu vaccines being developed at different places, such as the University of Texas and Texas A&M. I always forget which one it was. But remember, that was all part of the Blue Angel program, which is now morphed into all of the science that we have today, utilizing plants to produce human antigens to disease, the shape-shifting immune system that they've been talking about to be able to get into humans so it will protect you against any pathogen that's ever out there. She was a part of the CDC at that time. Well, right when Obama took office, she decided to step down, and she was the head of the CDC from 2002, ding, the year after 9/11, and 2009. She led the agency from one crisis to another, including the investigation into anthrax attacks that killed 5 people in 2001, the H5N1 avian influenza pandemic, the global outbreak of SARS, and the various outbreaks of food poisoning that, of course, happened all over. And in 2009, she decided to join Merck. So another revolving door is not just the FDA, the CDC as well, the ones who are telling you that little Johnny needs to get vaccinated because he ruined Disneyland vacations for all kinds of children not having that measles vaccine. Merck said in a statement, "We are very excited. Julie Gerbinger will be joining Merck where she can help expand access to vaccines around the world," says Gerbinger, who will head up the company's $5 billion global vaccine business that includes shots for chickenpox, cervical cancer, which must be the Gardasil, and pneumonia. So how frightening is that? And I don't really know much of what she's been doing since 2009. I remember in 2015, she sold a huge chunk of her stock, 38,368 shares to be exact, of Merck stock, valued at about $2 million. Says it was reported earlier this month, and this is in 2015, that Dr. Gerbinger, now executive vice president of the pharmaceutical giant Merck, sold 38,368 of her shares in Merck stock for $2,340,064.32. She still holds 31,985 shares of the company stock, valued at another $2 million. But besides examples like this showing a clear conflict of interest between government agencies tasked with overseeing public health and vaccine safety and pharmaceutical companies, the National Institute of Health also holds patents on vaccines such as Gardasil, and they earn royalties. You would have guessed it, ding, 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 from the sale of vaccines. Like, how unbelievably frightening is this? So, guys, anyone who ever doubted that we are just guinea pigs in a dish, don't doubt it anymore, because it's all about cash. So, all right, tomorrow I think we're going to twist our way somehow into brain technologies and the Internet of Things again. I think that's what I got lined up here. So make sure you stick around. It is Wednesday. Ms. Lonnie Clark is live up next. And then we have a live Josh with Anti-Tyranny News at 6 p.m. Eastern. The boys from New York at 8. Liam Schaff is a brand new show at 9 p.m. Eastern. Anti-Media Radio with Nick Burnaby at 11. All right, guys, have an awesome Wednesday, and I will get to you tomorrow.
You are listening to